Hello and welcome everyone. In this podcast we're going to talk about Mendel's second law, and that's the law of independent assortment. And again, that's his second law. Okay, as we get started in this podcast, I want to go through a description of Mendel's second law, his law of independent assortment. Following that, we'll go into a more detailed description by following a dihybrid cross. But let's start with his law. His law of independent assortment. We could define this law simply by saying that two different genes, you could also say allele pairs, but we'll stick with different genes, randomly assort their alleles during gamete formation. So if you remember when we took our genes, big R, little r, and the other one, big Y, little y, so two different genes, different allele pairs, and we crossed it to itself, this law complements Mendel's principle of segregation. So remember that Mendel's law of segregation says that these alleles, let's just look at R, that big R and little r, they separate during gamete formation, and you would expect 50% of the gametes to have big R, or little r. And remember, I'm putting in two of each because one diploid cell makes 4 haploid gametes. So you expect half of them to have big R, half to have little r. Same thing with the Y. When they separate, you'd expect half to go into each of the gametes. Now what this law of independent assortment says is that when this second allele pair, big Y, little y, segregate into these gametes, it's independent. Meaning big Y is just as likely to go into the big R gamete as it is just as likely to go into the little r gamete. When little y segregates away from big Y during gamete formation, it's just as likely to get paired with big R in this gamete or with little r in this gamete. So the key difference between the law of independent assortment and the law of segregation is that the law of segregation deals with one allele pair. 50% of the gametes big R, 50% little r. The law of independent assortment deals with two different genes, R and Y, and says that the second gene pair assorts randomly. It doesn't matter if it goes with big R or little r. It's random. And in explaining this, we're going to talk about a dihybrid cross. And if you remember from the monohybrid cross, that was a cross where there was just one trait that was being watched. A dihybrid cross follows two traits, or we could say follows two genes. So an example of a dihybrid cross using Mendel's P's might be looking at the two phenotypes of seed shape. So in this case, seed shape will either be round or wrinkled. And I'll draw that like so, as round or wrinkled. And then we'll also look at seed color. And seed color will either be yellow, which I'm going to use an orange marker for, because I don't have a yellow one, or it will be green. Yellow is the dominant color, and round is the dominant shape. The genes for round will be, or I should say the allele is big R, and for wrinkled it will be little r. For yellow it will be big Y. For green it will be little y. What we typically do when we assign gene letters like this is whatever the dominant phenotype is, that's what we follow. So we don't call this big R and this little W. It's always going to be the same letter. lowercase and uppercase for the wild type. So what we want to start off with is two strains that are pure breeding. The first one will be big R, big Y, big Y. So this will be a strain that is yellow and round. And we're going to want to cross that to a strain that is also pure breeding. That is going to be wrinkled and green. So little r, little y, little y. And when we cross this, what we're going to get is progeny that is all round and yellow. And that's because when we take this strain here and it makes seeds, each of the seeds are going to be big R, big Y. Because these two big R alleles, according to the principle of segregation, will separate from each other and each gamete will get a big R. Likewise, the two Y alleles, big Y and big Y, they will separate from each other during meiosis and will segregate into the eggs. So they will all be big R, big Y. Now when this other parent here makes seeds, all of the seeds will be little r, little y. Again, it's because these two alleles here, little r, they separate from each other during meiosis and they segregate into the gametes. Each gamete will have one little r. Likewise with the little y alleles, they segregate from each other and each gamete will get a little y. All the gametes will be big R and big Y from this parent, and all the gametes from this parent will be little r, little y. We know that all the progeny here will get a big R from this parent and a little r from this parent, and they'll get a big Y from this parent and a little y from that parent. So they will all be, we know they have to all be big R, big Y, little y. Since round is dominant and yellow is dominant, we know the seeds will be all round and yellow. So now we want to take these plants that all have round. So what we want to do next is take all of these plants that have round yellow seeds and cross them to themselves. And we call this self-fertilization. And we're going to go to the next board to talk about this. So we're going to take these round yellow seeds with the genotypes of big R, big Y, little y, and cross it to itself. And so we can take two approaches. We can take the approach of making a very large Punnett square, and that's what we're going to do first. And then I want to show you how we can use a method called the branch fork method. And I think you'll see using the other method is a little easier, because using a large Punnett square, it's easy to make some mistakes. So we know when this plant, actually each of these plants, because they're identical genotypically, when they make gametes, we know half the gametes are going to get big R and half are going to get little r. And so let's draw our gametes over here. And I'm going to draw them in circles like so. To remind ourselves, what we're looking at really always on the left or the top part of a Punnett square are the gametes. So we know when this plant here makes gametes, half of the gametes are going to get big R, and we know it's going to make 4 gametes, so let's go ahead and put big R and big R in 2 of these gametes. And so we know the other 2 gametes are going to get little r, little r. Again, this is just following meiosis. You know that with meiosis you always start with 1 diploid cell and you end up with 4 haploid cells. So that's all we're doing here. And we also know that half of the gametes are going to get big Y. And we know they assort randomly, so it's just as likely that this big Y is going to go in with this big R as it is that this big Y will go in with the little r. We would expect 25% of the gametes to be big R, big Y, and 25% of the gametes to be little r, big Y. Because of Mendel's law of independent assortment, these alleles, when they segregate into gametes, it's random. And for the same reason, we would expect little y to go in half of the gametes. So we would think that it's just as likely for little y to go with big R as it is to go with little y. Now since we're pairing up with the same individuals, we know up here we're going to have the same possibilities here. So let's rewrite all of these here up here for this parent. Okay, we now have our gametes on both sides of the Punnett square. And now, just like we did with the monohybrid cross, we're going to bring the gametes together and indicate what the progeny are below here. So we know here we're going to have big R and big R and big Y and big Y. So we'll just write big R, big Y, big Y. In fact, I would encourage you maybe now just hit pause on the tape and try to fill this in yourself, and then we can compare. But I'm going to go ahead and move forward here. And then we're going to have here, we're going to have a big R from here and a big R from here. And we're going to have a big Y and a little y. And on here we're going to have a little r, and we're going to have a from over here and a big R. And we're going to have big Y and big Y from both of these parents. Over here you're going to have big R and a little r, and you're going to have a big Y from this parent and then a little y from this parent. Over here you're going to have a big R from this parent and a big R from this parent, a big Y from this parent, and a little y from this parent. This one here, we're going to have a big R from both parents, and then we're going to have a little y from both parents, from here and from here. This parent, we're going to have a big R from this parent and a little r from this parent. Notice right now we're just looking at the genes and how they're segregating into the embryos down here. We're not talking about colors or phenotypes. Here we're going to have big Y and then little y. Down here we're going to have a big R, and then little r, little y. Up here we're going to have a little r from this parent and a big R. And then we'll have two big Y's. And then we're going to, in this slot here, we're going to have big R, big Y, little y. Here we're going to have little r from each parent, and then big Y from each parent, because they both are little r, big Y, little r, big Y. Here we're going to have two little r's, one big Y for this allele pair, and then a little y. Over here we're going to have a big R, big R from this parent, little r from this parent. Then we're going to get a big Y over here, and a little y. Big R, little y, little y. Here we'll have a little r, big Y, little y. And down here we'll have little r, little y, little y. So from this, we can now put in our phenotypes. We know this one's going to be round and yellow. Let's just draw it like that. This one we know it's going to be round and yellow. Here we know it's going to be round because big R, little r. Big R is dominant, and then big Y. So we know that one's going to be round and yellow. And again, we know this one's going to be round and yellow as well, because you're heterozygous for both, and the big R, big Y, the round yellow is dominant. Okay, here we're going to have a round yellow as well. Here we're going to have something a little different finally. We're going to have a round green. And here we're going to have a round yellow. And here we're going to have another round green. Here we're going to be round and yellow again. And same with this one. We'll finish off that column. Now we're going to have a round yellow again. And this one's going to be round and green. And this is going to be our first wrinkled one. So this is going to be a wrinkled yellow. Over here we're also going to have a wrinkled yellow. And here we're going to have a wrinkled yellow. And over here, the one and only we have, this wrinkled green. Okay, now we need to calculate the genotypic frequencies and the phenotypic frequencies. And so I need a little room up here to write, so I'm going to erase this very top part. So the genotypic frequency, this is the easiest one for this particular example. The frequency of big R, big Y is going to be 1 out of 16, because there are 16 possible choices here. In fact, I don't need to go through all this in each by each, because each of these are a different genotype. So the frequency of any one of these genotypes is 1 out of 16. Okay, so let's erase this. So now let's talk about phenotypic frequencies. And we have a few different possibilities. So let's write all the different possibilities we have. We have a fully dominant phenotype, which is round and yellow. And we also have a wrinkled yellow. We have a round green. And we also have a wrinkled green, like that. And now what we need to do is go through here and count each of the ones we have. So we have one for the round yellow. We have 1, 2, 3, 4. In fact, I think it's best to make a little check mark by each of these so you don't forget that you already counted them. So you have 1 here, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 of the round yellow. Now let's go back through and count the the wrinkled yellow. We have 1, 2, 3. We write 3 here. And then we have the round green. We have 1 here, 1 here, 1 here, and 1 here. So we have 3 of those. And finally, all by itself, we have 1 wrinkled green. So the phenotypic ratio is going to be 9 to 3 to 3 to 1. In fact, this is a famous ratio, 9 to 3 to 3 to 1. We will always see this ratio when we do a dihybrid cross where each of the parents are heterozygous. So we have 9 round yellow to 3 wrinkled yellow to 3 round green to 1 green wrinkled. So if you're given a dihybrid cross like this isn't too bad to do by the classic Punnett square. But there is another way we could have done it, and I'm going to show this other way, the branched fork method way, using a trihybrid cross. Where a trihybrid cross, to do it this way, would be a Punnett square that would have A up here and A below. Again, you could do it, but it takes a lot longer to do. And you have a greater chance of making a slight error in the combinations down here. So let's do a trihybrid cross using the branched method. Let's start off with a strain that is heterozygous at 3 different genes. Big A, little a, big B, little b, big C, little c, times the identical genotype. Big A, big B, little b, big C, little c. Now again, you can make a large Punnett square if you would choose. But let's look how we might do this using this branched method. Okay, so let's start the branch method by making a table. And what we're going to do is we're going to consider each of these allele pairs individually. So we're going to ignore the rest of them. If we had 4, 5, 6, we're going to ignore them all. So let's just look at big A, little a. We know that if you did a monohybrid cross of a big A, little a by big A, little a, that you're going to get 3/4 of those being big A. And you know, you're going to get 1/4 of these being little a. Alright, simple enough so far. Now let's look at the B allele. And we know that big B, little b times big B, little b, you're going to get 3/4 big B and 1/4 little b. Now, since it's just as likely that big B can combine with big A or with little a, we have to account for that here. So we'll say 3/4 big B could equally, just as equally, pair with the little a in the gamete. And so also we'll put the 1/4 little b here. Now what we want to do is consider our big C, little c. Now I'm going to start drawing some lines here so we don't get things mixed up, because this does get a little messy, but it's not too bad. Alright, big C, little c times big C, little c, we know that 3/4 of them should be big C, and 1/4 should be little c. I'm going to draw a line under my little c, because sometimes it's hard to tell the difference between a big C and a little c when we write them like this. So I'll put a line under the little c. Now, because big C is just as likely to pair with big B or little b, we know that we have to account for that here too. So 3/4 big C, 1/4 little c. Likewise, it's just as likely for big C or little c to pair with little a and big B or little a and little b. So we have to account for that down here. So we have 3/4 big C and then 1/4 little c, and 3/4 big C and 1/4 little c. Now to determine the phenotypic ratios of all these possibilities, we're going to multiply these through. So 3/4 times 3/4 times 3/4 is equal to 2764. So out of 64 possibilities, 27 of them will be big A, big B, big C. Now, this one, 3/4 times 3/4 times 1/4 is 9 out of 64. Will be big A, big B, little c. All right, now, again, big A, what's the probability that you're going to get a big A, little b, big C, and then big A, little b, little c. 3/4 times 1/4 times 3/4 for our big A, little b, big C, 9/64. And then 3/4 times 1/4 times 1/4 gives us 3/64 of a chance of being big A, little b, little c. And so now let's look at little a, big B, big C, little a, big B, little c. And so that's 1/4 times 3/4 times 3/4, 9/64. And then for the other one here, little a, big B, little c, 1/4 times 3/4 times 1/4. And that's going to give us 3/64. Now we want to look at these bottom two. Little a, little b, big C. Little a, little b, big C, which is going to be 1/4 times 1/4 times 3/4. And that's going to be equal to 3/64. And then our bottom one here is 1/4 times 1/4 times 1/4, which is equal to 1/64, being little a, little b, little c. So if you had to give a ratio for this, you would say it's a 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. I probably wouldn't ask it just that way. What I might ask, though, is to give you a hypothetical cross like such and ask you, what is the probability that you will have a big A, big little b, little c? So I might just ask this one alone. And so if you had this as a problem, you wouldn't have to fill all this out. All you would have to do is say, what's the probability of being big A? 3/4. Then you'd ask yourself, what's the probability of being little b? And that's 1/4. And probability of being little c is also 1/4. So you don't have to do the whole table if you're not asked to do the whole table. You can just do one component at a time. All right, now I'm going to erase this, and I want to provide one more example of something that would be absurdly difficult to do with a Punnett square, and would also be pretty difficult to do this way. But the way I asked the question, it's quite doable. And let me go over it with you real quick. And we'll do the practices of these in class too. What if I gave you this cross? I said big A, big B, little c, big D, big E, big F, little f, and I crossed that to a string that was big A, big B, big C, little d, big E, big F, little f. Now if I said, what is the genotypic or the phenotypic ratios of all the possibilities, that might not be very nice of me. So I wouldn't ask that particular question for a 6-character cross. So there are two different kinds of questions I could ask here. I could ask a genotypic one, that would be fair, and a phenotypic one. So for the genotypic one, I might ask, what is the probability of getting a genotype that is as such? Big A, big B, little c, big D, big E, and little f. So let's do that one first. And again, just approach this as 6 individual monohybrid crosses. And if you have to, you're welcome to do like little tiny Punnett squares all over the place. And we'll do a couple of them that way. Big A times big A little a. So we know big A, big A, little a. We know these are our possibilities. But what's the possibility of being big A, little a? Well, it's 1/2. Now let's ask, what's the probability of being big B, big B? Again, I would recommend maybe hitting pause on this and trying to do this on your own once you kind of get an idea of what I'm doing here, and then coming back. So these are our two possibilities here, big B, and big B, little b. And I'm asking what's the probability of being big B, genotypically. And so we'll fill out this tiny Punnett square down here, and we can see we have 2 out of 4 that are big B, big B. So we're going to go 2/4. And let's look at C. We have big C, little c, little c, little c, and we'll bring the gametes together. And again, we can see we have a 50% chance of being big B, little c. Now we'll do big D, little d. The probability of being big D, little d is 2/4. Now what about big E, big E? That the probability that one of them will be homozygous dominant is 1/4. Good. I'm glad we got something other than 2/4. That was getting a little tiring. Now we have 2 heterozygotes, big F little f times big F little f. Little f is going to be 1/4. So the probability that you're going to have this genotype from this cross, you just multiply this through. 2 times 2 is 4, times 2 is 8, times 2 is 16, times 1 is 16, times 1 is 16. So this is going to be equal to 16. Alright, so now let's multiply the denominators. 4 times 4 times 4 times 4 times 4 and times 4. And we get 4,096. On an exam, you could leave it just like this. You could also reduce it. I would accept either. And let's figure out a phenotypic ratio. Again, I'm not going to ask you to tell me all the possible phenotypes, but I could ask something like this. What's the probability of being big A phenotype, big B phenotype, little c phenotype, big D phenotype, little e phenotype, and big F phenotype? If you notice, I put a dash here next to the big A, next to the big B, next to the big D, and next to the big F. The reason I did that is because it doesn't matter what the other one is. Phenotypically, it won't matter if it's big A, or big A, little a. So in order to figure out the phenotype for like the big A here, we have to account for all 3 possible genotypes. Big A, big A, big A, little a, and big A, little a. So let's go ahead and walk through this. Again, let's do a couple with our Punnett squares. So if you have a big A, little a, and a big A, little a for our 2 alleles here for the Punnett square, and you bring the gametes together like so, what we see is we have 3 here that could be big A as far as the phenotype is concerned. So we'll write 3/4. Then same thing with the big B phenotype. A little different because we have 2 different parents on this one. Big B, big B, big B, little b. And now what you can see here is that they're all going to be phenotypically big B. So you could write 4 out of 4, or we could just make it 1 out of 1 times. Now let's look at the probability being little c, little c. Now in this case we do need to put both little letters there, because the only way you can have a phenotype of little c is if both alleles are little c. So over here we have little c as one of the parents, and then big C, little c. So let's draw a Punnett square here representing that possibility. And you can see from here that it's half of them. So we could write 2/4 here, or we could just write 1/2. That'll make our math a little easier. Now what's probability the phenotype is going to be big D? Again, you can see big D, little d, heterozygote times a homozygous recessive. And we can see that probability is going to be 1/2. Because this time we're asking what's the probability that the D allele will be present in its dominant form. So that's 1/2. Now how about little e, little e? Well, that's similar to this Punnett square. And we know to be fully recessive, there's a 1/4 chance of having that phenotype. So we'll go 1/4. And then our last one here is having a big F phenotype. And the probability of having a big F phenotype when you cross 2 heterozygotes, 3/4. And so if we wanted to know the probability of having a phenotype of big A, big B, little c, big D, little e, big F, we would multiply these numbers through. And when we do that, what we get is 3 times 1 times 1 times 1 times 1 times 3. We get 9. And down here, when we multiply these through, we get 4 times 1 is 4, times 2 is 8, times 2 is 16, times 4 is 64, times 4 is 256. Okay, that ends this podcast. Let's close with a short summary. In this podcast, we talked about the dihybrid cross. And we could also say multi-hybrid cross, where you could have 2 or more gene pairs interacting with each other, and how this law of independent assortment states that when you're looking at more than one gene, that the assortment of all of the different alleles is random. And we did some advanced crosses, where we were looking at more than 1 or 2 different genes, we were looking at 3, 4, 5, or 6. You should be comfortable doing the couple examples we did on this podcast and the ones we do in class. We talked about using the Punnett square to do these crosses, and how sometimes that seems like an acceptable way to do it. But we also talked about how sometimes we need to use this branched method, because it's going to get us to our answer much faster. All right, that's all I have for this podcast. If you have any questions at all, please make sure you come see me. I'll see you in class. Bye.